The arithmetic of weight management appears straightforward. Consume less energy than the body expends, and the body draws on stored reserves to compensate for the difference. The logic is consistent. The math holds. What doesn't hold, for a significant portion of the population, is the predictability of the outcome. Someone at 26 runs a 500-calorie daily deficit for 6 weeks and loses approximately what the calculation suggests. The same person at 43 applies the same deficit, same foods, same activity level, same duration, and the result is noticeably different. Slower, less consistent, sometimes negligible. This isn't anecdotal. It's reported broadly enough across clinical weight management literature to constitute a documented phenomenon. And what makes it worth examining is that the people experiencing it aren't making errors. The inputs are correct. The expected output simply doesn't materialize at the same rate. The common interpretation is that metabolism slows with age. This is partially accurate, but descriptively incomplete. It identifies the observation without explaining the mechanism, and the mechanism is where the more useful information sits. The standard response to a calorie deficit that stops producing results follows a predictable pattern. The deficit gets deepened. If 500 calories isn't working, the assumption is that the number needs to be larger. This tends to produce short-term movement but carries a ceiling. At a certain point, the body's adaptive responses become more pronounced and the efficiency of the approach decreases further. Exercise volume increases. More sessions, longer duration, higher intensity. The logic is sound in principle, but the relationship between exercise volume and metabolic output is not linear, and the additional expenditure is often partially offset by the body's compensatory reduction in non-exercise activity. Small, unconscious movements throughout the day that quietly decrease as formal exercise load goes up. Dietary protocols get refined, macronutrient ratios are adjusted, meal timing is restructured, intermittent fasting windows are introduced. Each of these modifications has a research basis, and each can produce meaningful results in the right context. But they share the same underlying assumption as the others, that the problem is one of inputs, and that adjusting inputs will resolve the output discrepancy. What this sequence consistently fails to account for is the possibility that the relationship between inputs and outputs has itself changed, that the processing system between them operates differently at 40 than it did at 25. The variable that dietary and exercise modifications generally don't address is metabolic rate at the cellular level, specifically how efficiently the body's cells convert available fuel into usable energy and heat. Resting metabolic rate does decline with age. The commonly cited figure is a reduction of roughly 1 to 2% per decade after the early 30s. But this aggregate number obscures a more specific mechanism. A meaningful portion of that decline is attributable not to reduced organ mass or muscle loss alone, but to changes in mitochondrial function, the same cellular infrastructure involved in energy regulation more broadly. Mitochondria are the sites where the body converts fuel, primarily glucose and fatty acids, into adenosine triphosphate. ATP is the form of energy cells actually use. When mitochondrial density decreases, fewer mitochondria per cell, and when the efficiency of each individual mitochondrion declines, the rate at which fuel is converted to usable energy slows. The body becomes, in a measurable sense, less metabolically active per unit of available fuel. This has a direct consequence for calorie deficit calculations. Those calculations are built on assumptions about how much energy the body expends at rest and during activity. If the underlying conversion rate has shifted, the calculation's output diverges from reality, not because the math is wrong, but because one of its foundational variables has changed. There is a compounding factor. NAD+, a coenzyme that participates directly in the metabolic pathways mitochondria use to process fuel, declines in concentration with age. Its reduction doesn't stop cellular metabolism, but it affects the rate and completeness of the conversion process, which affects how many calories are effectively metabolized versus stored, and how available that energy is for physical output. The result is a system where the same calorie deficit enters a different metabolic environment at 40 than it did at 25. The The arithmetic hasn't changed, the biology has. The research response to this phenomenon has produced two distinct directions. The first is exercise-based. Resistance training, in particular, has documented effects on both mitochondrial biogenesis, the generation of new mitochondria, and on the preservation of muscle mass, which is itself a driver of resting metabolic rate. This is well established, and it explains why resistance training recommendations for adults over 35 have become progressively more prominent in mainstream health guidance over the past decade. The second direction is molecular. Researchers in cellular biology and aging have investigated whether the NAD+ decline that accompanies aging can be addressed through precursor supplementation, introducing compounds upstream in the pathway through which NAD+ is synthesized. NMN and NR are the most studied candidates in this category. The distinction between these two directions matters. Exercise-based interventions work primarily through volume and mechanical stress. They stimulate the body to generate more mitochondria and preserve metabolic tissue. Molecular interventions, where supported by evidence, operate on the efficiency of existing mitochondria and the availability of the coenzymes they depend on. These are not mutually exclusive, but they address different parts of the same underlying problem, which means deploying one without the other leaves part of the mechanism unaddressed. The commercial category that has emerged around NAD+ precursor research is worth understanding on its own terms, separate from the marketing language surrounding it. NMN and NR supplements are not positioned in the research literature as weight loss products. Their proposed mechanism is not calorie reduction. What the research investigates is whether restoring or supporting NAD+ availability affects the rate and efficiency of cellular metabolism, which is a different claim operating at a different level of the system. For individuals who are managing calorie deficits and finding the results inconsistent with what the arithmetic predicts, the cellular metabolism question is the relevant one. Not whether they are eating correctly, but whether the downstream processing of what they eat is operating at the same rate it once did. Products in this category vary considerably in formulation quality, dosage transparency, and third-party verification. The research basis for the category exists, but it is still accumulating, and the gap between what studies show and what product marketing claims is often significant. That gap is worth evaluating independently. The calorie deficit model is not wrong. Energy balance remains the foundational principle of weight regulation, and the evidence for it is not in question. What the model doesn't capture is the age-related variability in how that balance is processed at the cellular level. The same deficit applied to a different metabolic environment produces a different result, not because the person is doing something incorrectly, but because one of the system's core operating parameters has shifted. Understanding that shift doesn't change the inputs, it changes the questions worth asking about them.